Good morning, children. So let's continue discussing the different methods of separating the components of a mixture. So in the last class we have discussed about the process that is the sublimation. So today we'll discuss another one method which is known as chromatography. So chromatography. So in this process different components of a mixture are separated due to their differential movement in a stationary solvent. So in this process the different components of a mixtures are separated due to their differential movement in a stationary solvent. So the applications used to separate colors of a dye and used to separate drugs from blood, as we can see in the picture. It consists of a filter paper. So where we marked a dye, as you can see in the first diagram. So there is a drop of dye and it is inserted in such a manner in the solvent that the dye is just touching the solvent. So as we know, the dye doesn't consist of a single color, it consists of many colors. So we insert that strip of filter paper in such a manner that the solvent is just touching the drop of dye. So in the second picture, as we can see, as the solvent gradually rises up in the strip, so the colors are separated. As we can see, the first that red color is obtained, but the orange color moves further than the red color. So just above the red color, orange color is obtained, and similarly just above the orange color we obtain the yellow color. As the different components, as the mixture consists of different components, so we obtain the different components at different levels. That means at different height we obtain the different components. So this method can be used to separate colors of a dye. Similarly, it is also used to separate drugs from a blood. So if the blood contains drugs, that means the blood, other components of the blood will remain at the lower level, and the drug samples, that means the drug components which are actually present in the blood, it will be available at the higher level. So similarly, it can be used to separate drugs from blood. So the next process is distillation. So in this process, mixture of two or more miscible liquids can be separated that have different boiling points. So applications used to separate acetone and water. So in this process, mixture of two or more miscible liquids can be separated that have different boiling points, as we can see in the picture. So this is the process of distillation. So as we can see in the flask, so there is a mixture of acetone and water which is heated. As it is heated, the boiling point of acetone is less, that is 56 degrees centigrade, and the boiling point of water is 100 degrees centigrade. So when we heat it at the temperature of say 56 degrees centigrade, so there is a thermometer on top, as you can see, to measure the temperature. So when we heat up to 56 degrees centigrade, the acetone starts boiling and the vapor of acetone rises. So as it rises up, it passes through the tube which consists of a condenser, that is water condenser. That means the cold water is being pumped there. So as the cold water is being pumped there, when the vapor reaches that condenser, it condenses. That means again it turns to liquid state, and hence it is collected in the vessel. So in such a manner we can separate, that is acetone and water. So distillation, but one point is to be noted, that in distillation, as we can see, the boiling point of acetone is 56 degrees centigrade and the boiling point of water is 100 degrees centigrade. So this process of distillation is only used where that means the difference in the boiling point is more than the, usually if it is less, another method is followed. But if the difference in the boiling point is more, since one is 56 degrees centigrade, so and the other is 100 degrees centigrade, so there is a huge amount of difference in their boiling point. So in such cases we use the process of distillation. So next is the fractional distillation. That means this process is only used where the difference in boiling point is less. Earlier, only the distillation process we used where the difference in the boiling point is more, but it is used in the case where the difference in the boiling point is less. So in this process, the mixture of two or more miscible liquids can be separated that have different boiling points, similar as above. So the applications of fractional distillation is the separation of oxygen, liquid nitrogen, argon from air. So it is used to separate crude oil components like petrol, kerosene, diesel, jet fuel, tar, etc. So in these fractional distillations, as we can see in the picture, similar as above, it consists of a flask which consists of a mixture, and on top of that one fractionating column is being attached. One fractionating column is being attached and one tube, and the rest of the thing is similar as the distillation process, that one tube is being attached to that fractional column and that tube passes through the water condenser, and there is a vessel to collect the component. So when we heat the mixture, so what happens, suppose it consists of two mixtures, that the boiling point of one mixture is say 50 degrees centigrade and the boiling point of other mixture is say 55 degrees centigrade. So when we heat the mixture, up to 50 degrees centigrade, then the boiling point of that component which is 50 degrees centigrade, it rises up as vapor. As it rises up as vapor, so it enters the fractionating column. The fractionating column actually consists of beads of glass. Some beads of glass are there. So as it rises up, similarly the other one is also, the boiling point is very less, so it is 55 degrees centigrade, so it also rises up and enters the fractionating column. So the boiling point of that component whose boiling point is actually less, that is 50 degrees centigrade, it will pass through that beads of glass, but the boiling point of that component whose boiling point is more, that is 55 degrees centigrade, as it enters the fractionating column through the beads of glass, it condenses there and again fall back to the flask, that is distillation flask, and hence the component whose boiling point is 50 degrees centigrade, it passes through the tube and hence through the condenser, and at last it is being collected in the vessel. The component is being collected in the vessel. So this method is actually followed where that is the difference of boiling point is very less, and the rest of the, only one thing is different from distillation, that is a fractionating column is being attached there, and the rest of all the components are same as distillation. So applications of fractional distillation, so it is the separation of oxygen, liquid nitrogen, argon from air. So that means the air is first compressed and cooled by increasing the pressure and decreasing the temperature. As we already know, that means when the air is compressed by increasing the pressure and decreasing the temperature, it will liquefy. So that liquid air allowed to warm out slowly in fractional distillation column. So in that fractional distillation column, we will obtain the different components of air, that is oxygen, liquid nitrogen, argon, etc. And the second one, it is used to separate crude oil components like petrol, kerosene, diesel, jet fuel, tar, etc. As we know that the crude oil that we obtain from the underground, it consists of different kinds of components. Altogether it consists of petrol, kerosene, diesel, jet fuel and tar, etc. So using this process of fractional distillation, so there are these are not like this laboratory fractional distillation. So it consists of a very huge fractional distillation column where the different components of crude oil, that is petrol, kerosene, diesel, jet fuel, tar, etc. are separated and it is obtained from there. So the next and the last process is crystallization. So in this process, impure mixture first dissolved in a suitable solvent, then crystallized one component, used in purification of water and used in separating alum and copper sulfate. So as you can see in the picture, for example, we take some seawater in a glass. As we know that it consists of different kind of impurities. That means if we allow to rest the seawater for some time, all the impurities will settle down at the bottom, and now it consists of salt and water. So in this case, we'll enter one thread in the glass of water and lower the temperature gradually. So if we lower the temperature gradually, what will happen? The crystals of salt will form on the thread, as you can see in the picture. So crystals of salt are formed on the thread. So in this manner, that salt can be obtained. The crystals of salt can be obtained from the seawater. And applications, it is also used for the purification of water. This crystallization process is also used for the purification of water. And it is also used in the separating alum, so, and copper sulfate. So that's all for today. The remaining we will discuss in the next class. Thank you.